Hello, Manchester. God, you guys are so quiet. Same thing happened last year. It's time for This Week in Microbiology. This is episode number 53, and today is March 27th, 2013. Hello, everyone. I'm Vincent Racaniello, and you are listening to TWIM, the podcast that explores unseen life on Earth. Today, we're coming to you live from Manchester, England. We are at the spring meeting of the Society for General Microbiology, and we're in front of a live audience. I've collected some of the speakers from today's session, and we're going to talk about next-generation antimicrobials. The purpose of this podcast is to bring microbiology to the world, and you guys out here in the audience are watching us, but many thousands more are going to hear this eventually. And this gives them an opportunity to learn about the cool things that we do. Podcasts make science engaging and understandable. Before I get started, we need to tell you what the weather is here in Manchester. Unfortunately, it's been very cold and windy and snowy this week. And it's currently about 3 degrees Celsius. I think it's cloudy out, right? Yeah, I didn't see any blue sky. Last year in Dublin, it was shirt sleeve weather. So Manchester, you really got to do some catching up. I've got a co-host today. Now, usually I have Elio Schechter and Michael Schmidt joining me on TWIM. So I've recruited a co-host to help me out. She's a professor of microbiology at the University of Birmingham, Laura Piddock. Welcome.
Thank you.
Thanks for joining me, and you came back especially today on the train to do this.
I did.
Appreciate it. You're going to stick around, I'll buy you a beer later.
Thank you.
All right, make up for it. Also joining us today over on Laura's left from the University of Nottingham, Paul Williams. So I feel like a talk show. Well, in fact, that's exactly what this is. On my right, professor of bacteriology at the University of Wisconsin-Madison, Kalin Vetsigian. Vetsigian?
Yes.
Anyway, thank you very much for joining us. I am familiar with mispronounced last names.
Me too.
Can you pronounce my name?
No.
No. Okay. And finally, on Kalin's right, Chief Scientific Officer from Amplify Biosciences Corporation, David Harper. Thank you for joining us as well. And as I said, I've picked these participants to kind of echo today's theme, which has been next generation antimicrobials. And I wanted you to kick us off, Laura, because you have a lab and you do research, but you also do some outreach, which is very different. So I wonder if you could tell us about that a bit.
Okay, so I work at the University of Birmingham, as you've just said, and I have a research team where we're primarily investigating the mechanisms of antibiotic resistance. We look at 2 types of resistance: multi-drug resistance conferred by efflux pumps, and we also look at plasmid-mediated resistance and try and understand how plasmids move around from bacteria to bacteria. And we then use the information we discover, particularly with multi-drug efflux pumps, to try and think of ways in which we can counteract their activity, because the efflux pumps are responsible for inherent drug resistance in most gram-negative bacteria. But these pumps can be overproduced and cause clinically relevant levels of resistance in the drugs that we actually want to use. So we're using basic science to sort of further our understanding as a platform for drug discovery. With my scientific background, a few years ago, I became president of the British Society for Antimicrobial Chemotherapy. And when I was at an SGM meeting at Harrogate, Professor Ian Chopra, who was at the University of Leeds, came up to me and said, well, what are you going to do about antibiotic resistance and the lack of new drugs? I was a bit surprised, but went back and talked with the Council of BSAC, and we ended up saying we were going to try and enter this area and find out what all the problems were, along with activities by IDSA and REACT and other groups around the world. But the key finding we had was that there was a lack of public awareness of the issues of antibiotic resistance and the lack of new ways of treating infection, not just antibiotics. So we launched Antibiotic Action 18 months ago, which is a public awareness initiative throughout the world. And we've been very, very active, as you know. So lots of talks, lots of TV work. So we're talking not just to microbiologists, infectious diseases physicians, but the general public, politicians, and like-minded groups in other countries.
So why is it important to tell the public about that? I mean, I know we have to tell physicians and drug manufacturers, but why the general public?
It's important to tell everybody about how important antibiotics are and how they underpin most areas of medicine, and so that they understand when they should be used and, more importantly, when they shouldn't be used, because we need to preserve their activity and use them properly. And it's really important they understand how easy it is to share antibiotic-resistant bacteria, and we want to stop that so that we preserve our drugs. And we also want the public to understand that there's other ways that you can prevent infection or reduce infection.
Mm-hmm. Do you also talk to farmers to try and get them not to use antibiotics?
Very much. In fact, in 2 weeks' time, I'm talking at a farmers and veterinary science meeting. So, yeah, and one really important thing is quite interesting. People try to blame other groups. We've got a lot of antibiotic resistance because there's too much use in animals, and then the vets will say, oh, well, there's too much use in humans, and then somebody else will blame another group. The reality is we're all responsible. We all take antibiotics, whether we're ill or whether we give them to patients if we're a physician. Scientists, we use them in our labs. We undermine their effectiveness by using them so much. So we all have to play a role in understanding how to use them properly. Because when we have new treatments, as I hope we will do, one way or another, we have to then use those in a very different way to the way we've got used to using antibiotics now.
So what is one message we could send to everyone who's listening today to improve their perception of antibiotic use?
To recognise that antibiotics save lives, they're miracle drugs, and to try and get people's perception of antibiotics to be similar to when people think about anti-cancer drugs. They're very valuable. The difference is, of course, cancer drugs cost a lot of money, but antibiotics don't. We don't value them in society in the same way, and we really should.
So you're doing a lot of work, and your organization does, but I think microbiologists in general also need to step up, right?
Absolutely, and we really want to encourage everybody to work with us. So Antibiotic Action has a global cohort of what we're calling Antibiotic Action champions, and they're people just like me, like you as well, go out and talk to everybody we can, spread the message of, you know, that antibiotic resistance is a serious global issue, and it affects everybody. It will affect us eventually. We could suffer by not having effective treatments, and we want everybody to spread the message.
So here on this podcast, TWIM, we often talk about it, but I think others should do the same and try and tell the public that it's really important that you view these, as you said, as essential to our health. All right, with that little beginning, let's start with you, Paul. Now, I noticed the title of your presentation, which was this morning, is Quorum Sensing of a Viable Antimicrobial Target. So I thought it would be interesting to have you join the conversation. Maybe you could tell us about that. But first, tell those who don't know, what is quorum sensing?
Okay, well, I guess quorum sensing is a very nice term which has really stuck, because it's one of those ways which captures a phenomenon that for many years we didn't really appreciate existed in bacteria. Now, of course, bacteria are single-celled organisms. They divide by binary fission, so 1 becomes 2, 2 becomes 4, and so on. And so they possess just about everything they need to reproduce and behave. And so we didn't really ever think that microbial cells had any reason to communicate with each other. So quorum sensing is the nice term that has actually grown with us to describe the way in which bacterial cells communicate with each other in order to do something together, collectively. So in essence, it's a mechanism for controlling gene expression at the level of population. So how do bacteria talk to each other? Well, the simplest way is to make a small chemical that you release that the neighboring cells can pick up, and collectively together they can take a census of their numbers, basically by titrating the concentration of signal molecules there. So the idea of quorum sensing and the quorum bit, of course, if you had a company and a board of a company needing to make a big decision, they were going to take over the Apple Corporation, for example, then you need to make sure that that board was quorate, i.e. enough voting members were there to make that decision binding. And that's really what quorum sensing is all about in bacteria. It's bacteria ganging up together to make a collective decision about what they're going to do next.
All right, and you think you can target this as an antimicrobial approach?
So in the context of my work, I mean, a lot of the work has been, first of all, to understand what is it that bacteria control using quorum sensing. What's the nature of the chemical signal? What does it actually do? What does it switch on and off? And the two areas that became really very exciting was one, that bacteria use quorum sensing to control their virulence. So that's to control their ability to cause disease. They also use quorum sensing to control, for example, biofilm formation and development, which of course is a major problem in most infections, where bacteria become these sort of slime cities, which stick to surfaces and are very difficult to eradicate, tolerant to most conventional antibiotics, and a major problem. So if you think of quorum sensing as the bacteria deciding to make a decision about when to behave badly, so if we're thinking about reforming them, rather than trying to kill them, which of course is always the problem when you're developing and thinking about antibiotics, you're thinking about agents that kill. And of course, agents that kill generally select for resistance. If instead of killing them, you actually stop them behaving badly, the selective pressure is not as great. And therefore, you may well have agents which don't encourage the development of resistance. Right. So in understanding the basic molecular biology of a quorum sensing system, you can begin to see how you might develop agents and therapeutics that might control the infection by, for example, taking out the signal molecule. So you might have an enzyme that destroys the signal molecule, or an antibody that captures it so they can't signal to each other anymore. Alternatively, obviously the bacteria have to sense and respond to that signal molecule. And so one way, of course, in which they do that, there are surface receptors. And so if you, in a classical pharmacological sense, design a receptor antagonist, then you can block the activation of virulence by blocking the ability of the organism to sense that signal molecule. And then thirdly, of course, the bug has to make the signal molecule. So that's also another target that one might go for. So you've got several different ways in which you could interfere with the ability of bacteria to communicate and to cause the infection.
So it sounds to me that sensing is not essential for bacterial growth, right?
No, sensing is not essential for bacterial growth. So if you take a nice rich medium in a lab and you take a mutant that can't sense, then it will grow perfectly okay. But of course, when we're thinking about targeting virulence and the ability to cause disease, the growth environment is not a nice meat soup. It's inside a higher organism with an immune system, and of course that organism has to adapt. It has to deploy all of those toxins, surface adhesins, and virulence factors that allow it to establish and develop infection. So it may not. So if you block quorum sensing, it can't deploy virulence factors, and so it may in fact not grow, even though these agents would not be targeted towards growth per se.
All right. So the idea is if you targeted factors that control virulence, that would be enough to control an infection. You don't actually get rid of the infecting agent.
Well, no, you would. Yes, of course. So one of the issues would be that you would probably find that such molecules may not work terribly well in an immunocompromised individual, and of course they may work even better synergistically with a conventional antibiotic.
Right. Right. So you're depending on the immune system to get rid of the infection.
To get rid of the organism,
right? By reducing the virulence. Okay.
How have you got into translating the basic research that you've just described into something that's more treatment-like? You've done some stuff in animals.
Yeah.
And how optimistic do you feel of actually translating the research into something that eventually could be used to treat an infection rather than prevent an infection?
Okay, so of course you have to have these sorts of molecules in the first place, and that means actually setting up the chemistry that actually goes along with the microbiology and the infected animal models and everything else to actually get to a point where you can see that you can attenuate the infection. And so, of course, mostly with this kind of research, you're starting several layers back, and that initially you're looking to develop a molecule which will block quorum sensing in a culture. And so, of course, there you're using all of the molecular biology skills that you've got. to develop an agent that you then pharmaceutically would have to develop. And of course, in most academic institutions, the developmental part comes next. And so that means you have to convince a company usually. And one of the problems in terms of any novel therapy, novel target when it comes to the industry is they really rather like things that kill. And so if you have something that attenuates, they're not terribly comfortable with it. So even though there are clear advantages when you think about issues like the development of resistance, there are disadvantages in that, lots of different bacterial species don't all speak the same language, and therefore the same single molecule inhibitor, the same QS inhibitor will not hit all organisms at once. So then you've got to choose your disease. And I think you've got to focus in on particular organisms where it may be beneficial to target that quorum sensing. In others, it may not be. So organisms like Pseudomonas aeruginosa and Staphylococcus, where there's a very clear pathway to the control of virulence through quorum sensing. You can begin to use the quorum sensing signal molecules themselves as the template for generating an inhibitor. So Staph aureus is a good example, where different Staph aureus strains actually inhibit each other. There's a lot of cross-group inhibition because one Staph aureus will make a molecule that's slightly different to another. While it activates self, it will actually inhibit non-self. And so that gives you a platform to begin to develop those drugs. Now, what we've seen so far with a lot of the compounds that we've made, which are effective in culture-type assays, when you start to get in vivo into your animal models, in superficial infections, these molecules often will work. But once you go systemic, the problem with many of the molecules we've developed so far is they're very hydrophobic. So pharmaceutically, their bioavailability is a problem. And that again is where, the drug industry with its drug development pathways, it's got to be bioavailable, it's got to get to the right place. If it gets sequestered around the body and isn't available for killing the organism, then it's a problem. So there's a whole raft of pharmaceutical drug development processes that once you've got your lead compound, you then really have to develop that further. And that isn't been traditionally something that university labs have done. I think that may come because, as pharmaceutical companies are changing the way in which they operate, they are looking to academia to perhaps provide more of an input into the later stages of drug development in terms of the sort of chemistry, the ADMET, everything else that you need to do before you go into man. That's something that we haven't traditionally done, but it's somewhere that we may end up going.
I think really you've picked up on two very important points really for academic input in this area. Particularly, there's quite a lot of different virulence mechanisms that people would like to use as a way of treating infection. And it's really persuading hearts and minds that this would be a good strategy, and persuading a physician to use a molecule or treatment that doesn't actually kill the infecting organism. It is going to be quite a challenge. And the second one, as you quite rightly point out, is having a good target or even good inhibitors doesn't necessarily make a good drug. And translating from molecules in the lab into something that has good pharmacokinetics, good pharmacology, isn't toxic, is a major challenge. And again, the expertise in industry is there, and we've got to now find a mechanism to start linking up academia, small, medium enterprise SMEs, and big pharma, so we can actually all exploit our skills to be much more efficient than just relying on pharma to do everything, which is what's historically happened.
Sounds like you have some candidates already that you've tested to a certain point in animals, right?
Yes, we have gotten to a certain point, but what we tend to find is that they look great in the lab, and once you get into tissues, you start to find that the efficacy goes down. So we really need to refine the chemistry to actually get the right sort of pharmacokinetics, pharmacodynamics. The dosing's got to be right. There are lots of things that one needs to do before they get anywhere near.
Does resistance to this quorum sensing inhibitors arise in the lab?
Sorry, I missed that.
Does resistance to these molecules arise in the lab?
So one of the questions as well people often ask is we assume that because the compounds don't kill the organism, that there is less of a selective pressure to develop resistance.
Actually, you might expect even the opposite, selection against resistance. In some cases, if you imagine
in
that
some cases,
these guys cooperate and pathogenicity comes through cooperation, if you block their quorum sensing, in a sense, they're not investing in making these small molecules, and then if a resistant mutant arises, then it will regain its cooperation, but there will be no one to cooperate with. So in these situations, you would expect actually there to be selection against resistance, but I'm wondering whether we can hope to have a drug for which there will be no resistance. Is this just theoretical kind of abstraction?
Again, it's one of those things that people have only just begun to look at, because there are not very many quorum sensing inhibitors out there, and some of those that have been worked on for a number of years, like the furanones, one of their problems is that the sort of therapeutic index, the dose at which they start to inhibit growth versus the quorum sensing inhibition is quite close and quite narrow. So when you're trying to select for resistance, if you up the dose, what you're doing is actually selecting for resistance towards the growth inhibitory properties, not the quorum sensing inhibitory properties of the molecule. But of course, I think with any drug that you throw at a microorganism, if it doesn't like it, and if it's in the right place. So if the organism can't grow in vivo because you've blocked it through quorum sensing, then you've got a selective pressure there for it to actually overcome that. So I think whatever you throw at a microorganism, I think it'll find a way around it eventually. It just depends on how long that period is.
But there's a similarity here, isn't there, to another next generation approach, which is monoclonal antibody fragments, because there's one in the clinic now which blocks a toxin secretion pathway and doesn't appear to lead to resistance but doesn't kill. So again, you're relying on the natural processes of the immune system and clearance in the body to actually resolve the infection. What you're basically doing is shutting down the virulence. So that it has a very similar approach from where I'm sitting.
Paul, we talk about molecules exchanged among bacteria. How about between bacteria and their eukaryotic hosts? Are there any?
Well, there's a whole story, for example, of bacteria sensing neurotransmitters like noradrenaline, the catecholamine compounds. There's a number of instances where bacteria seem to respond to cytokines. So there's a nice story with Pseudomonas that you see upregulation of quorum sensing in response to one of, I've forgotten which one of the cytokines, but there's a nice pathway that does that. So there's a lot of exchange, I think, between the two. Sometimes that exchange leads to upregulation of quorum sensing, or even potentially downregulation. So with the catecholamine neurotransmitters, there certainly appears to be a stimulation of bacterial growth through a signal transduction pathway that's in some ways similar to a quorum sensing pathway.
So could these be targets as well?
Of course, they've got other functions, other physiological functions, so you probably wouldn't target those, but you might target the bacterial response to those molecules. In fact, there is some nice work from a group in the States where in fact they've focused in on one of the receptor sensor kinases, which normally responds to, I think it's noradrenaline, and they did a nice chemical library approach, came up with a compound, and then screened it, found an inhibitor, and it seemed to work reasonably well in animals.
So give us a sense of the size of the labs working on this. Is it a few, or is it many, or?
It's. I think the whole of as the quorum sensing field has expanded hugely, and I think it's really, the term was only coined in about 1994. So there are papers before then, but we didn't call it quorum sensing then. So once quorum sensing was stuck, and if you kind of do a literature search, you'll find an exponential growth over the last 20 years, just about, of papers on quorum sensing. And if you then look at quorum sensing inhibitors, you'll find that equally there's an exponential rise in people looking for those sorts of compounds. And largely that's been possible because many groups, including my own, have developed biosensor type approaches for sensing the signal molecules themselves. And of course they lend themselves quite nicely to screens for inhibitors. So it can be switch on of color, switch on of light, switch on of fluorescence, and you switch it off. So the literature is now full of those sorts of papers. But often the molecules have come from a variety of sources and are often not taken any further at this point.
Okay. We have a question.
Is this on?
Yeah.
Oh, it's on. We have a question from a Twitter user. Ronan McCarthy asks Professor Williams, how long do you think we are from a quorum sensing inhibitor being commercially available for use in humans?
A very long way. Could be at least 20 years, I would suggest.
Wow.
Possibly more.
Any others? That's it?
There's one other question from one of our online viewers, an anonymous user, who asks, it says, improving awareness of the problem is admirable, but there have been similar long-running campaigns in Europe and the US, run by the RDSA, for example, and yet nothing changes. What will it take to achieve some real action?
Okay, so those campaigns I'm well aware of, and in fact we work closely with them, particularly IDSA and REACT, and there's APUA and there's WAAR, a French-based group. The difference between antibiotic action, and if you want to look it up, it's antibiotic-action.com, is that we aren't speaking to the converted. We're not talking to microbiologists and infectious disease physicians primarily. We are, obviously, because we're here today. We're going out and talking to everybody. And our champion network, which is global, includes people from all walks of life and all ages, from very young people to quite old people. And that's what it's going to take. We have to view it that we have to mobilise everybody to understand the nature of the problem and how serious it is. And that has not happened with any of the other groups, and that's why it's been picked up so much more, because so many more people are now talking about it. And I view it as a wave. It's been a little bit of a ripple, you know, to put the stone in the pond, and now the ripple is getting out and it's turning into a bigger and bigger ripple. And there's been such a lot of activity in the last year. You know, we've only been going since November 2011, and there's been a lot of activity, particularly in the last month or so.
All right, let's turn to Kalin. Did I get it right?
Yeah.
Kalin.
Perfect.
From Bulgaria, right?
Yeah.
I'm not. I'm from the United States. I'm of Italian descent. My name is Racaniello.
Racaniello.
Okay. Now, the title of your talk that caught me is Antibiotic Inhibition and Attenuation in Soil. So you like to play in the dirt. And I read one of your papers, and you looked at the microbes on 4 grains of soil.
Yes.
Tell us about what you're doing.
So I'm interested in evolution of biocomplexity, and I started in theory, but at some point decided there is not enough data, and decided to take some data myself on the statistical properties of microbial interaction networks. So one thing I've done is just isolated some bacteria from different grains of soil, really small particles of soil, like 200 nanograms, and isolated many antibiotic makers from these different grains, and started looking at, measured all the pairwise interactions among them, like big 64 by 64 matrices. And I was looking at, first of all, what are the basic statistical properties, like what's the probability that if you isolate, that if you grab 2 bacteria from soil, one will inhibit another. And it turns out that among the antibiotic makers, this probability is as high as 50%. So it's a really dense network of inhibitions. And then you can ask, okay, if you really have such a dense network of inhibitions, then how do these communities coexist? And it turns out that that at the same time they're also kind of protecting each other through collective resistance mechanisms. So they degrade each other's antibiotics at equally high frequency. So you have a situation where many soil microbes produce many antibiotics, and then they also degrade many of these antibiotics. So it's kind of a really interesting situation to study theoretically. And we've actually managed to, so we've derived some statistical properties from these experiments, including reciprocity. So bugs that are isolated from the same grain of soil are more likely to exhibit reciprocal interactions than bugs from different grains. And we managed to reproduce all these statistical properties with an ecological model. But it's a very interesting model in the sense that the only way we could reproduce these statistical properties is by actually putting evolution in the model. So if you turn off evolution, if you imagine that communities are just evolving through some ecological interactions, we could never get the matrices right. And it seems that the fact that they're evolving, which in these communities can happen through the process of horizontal gene transfer, that actually enabled us to reproduce the statistical properties. So it's kind of a new way, ecological way to look at complexity. Typically, when theoretical ecologists at least think about complexity, they think in terms of ecological stability. That means, imagine there is no evolution. Are these communities stable? And what this theoretical work suggested is that natural communities might be stable, might be diverse not because they're ecologically stable, but because they're excitable in the sense that even a little bit of evolution is sufficient to pump up a lot of diversity there, and there is always a little bit of evolution happening. So, yes, another way to think about this work is that I've been kind of, which relates more closely to what we've been talking about, the problem of resistance, is just to ask whether antibiotic producers in nature are also feeling the problem of resistance, right? Is it just us in the clinic trying to always come up with new antibiotics, or in natural communities, antibiotic producers also need to, over evolutionary time, to change their production capabilities, right? And for example, it's known that there is very fast coevolution between phages and bacteria in the ocean, but the question whether there is equally fast coevolution between antibiotic production and resistance in, say, soil environments is not so well explored. So I've been interested in that, and as part of this work on looking at microbial interaction networks, we, of course, also looked at the relationship between the network properties and phylogeny, and in particular, we asked if you are more similar isolates having more similar production profiles, and it turns out that the answer is no, which means that antibiotic production seems to be evolving very fast, at least relative to the time scale that we had, which is the rate at which a barcode gene code 16S is changing. So we don't know how fast it is, but at least so far the data seems consistent with the idea that there is a constant kind of cat and mouse game between production and resistance, and I think it will be very interesting to try to figure out how exactly do soil microbes, or Streptomyces in particular, change their production capabilities. Do they tweak with their antibiotics? Do they play with the regulation of the antibiotics they have? And we know that some of them can make 20, or even in some cases 40 different small molecules. Or do they just play with the combinations of antibiotics they have? And I guess we can answer some of these questions by sequencing and things like that.
So basically, in the soil, what you're saying is that the community remains stable. There are no winners. So one species doesn't wipe everything else out by producing antibiotics, right? There's a steady state cooperativity of some kind.
Yes, there is state, but dynamic steady state.
Right. Now, is this because it's better to be diverse as opposed to have one species dominating? Or I don't know if you know the answer to that.
Well, I mean, it's just like with, I guess, with the computer viruses, right? Once you have one dominant guy, all the viruses got over this. I mean, or all the antibiotic inhibition. But so I don't know whether it's better to be diverse or not. It's just
that's the way it is. Yeah.
If you actually it's hard to think about this process. That's why we do computer simulations, and that's what's happening in computer simulations is that there are instabilities towards diversities. Yeah. You can think, for example, that if you have one species in one niche, and you can ask, is this situation stable or not? And the answer, it's not stable with respect to the evolution of production and resistance, because if a production gene, if a production cassette comes from somewhere, then you start invading, you start gaining advantage because you will be outcompeting the sensitive strains, but then resistance will come along, and then the resistance will be outcompeting the sensitive, and then you have a paper scissors roll game. And this means that the dynamics is unstable with respect to emergence of production resistance, and I think that's what drives, one of the factors that drives the diversification in, let's say, antibiotic production and resistance in soil.
So you have to do this with models, because in the lab you study Streptomyces, right? One species, but in fact in the soil there are many.
Yes.
So you can't study that readily, so you model it. Is that the idea?
I mean, I do two things in my lab. One is study natural communities with these statistical approaches, and then because we really want to model things and understand things in details, we are also starting to play with toy communities in the lab, where instead of 10,000 bacteria, we put 10 or 5, and then we can really nail down, hopefully, what are the processes that are going on. But even if, with respect to the natural environment, the goal of obtaining statistical data is that it allows us to, this is data that we can use to discriminate between different ecological models. Right? That's the only way to discriminate between those. And these ecological models, in fact, constitute hypotheses about what's happening in nature. For example, the model that we did before, kind of, the hypothesis there is that you need evolution in order to maintain diversity, and if you somehow can cut genetic exchange, let's say by curing plasmids or something in an artificial community, then diversity will be collapsing more rapidly. So you can test these things subsequently.
There's a remarkable figure in one of your papers where you compared resistance in the clinic, resistance to antibiotics in the clinic versus the soil. In the soil, it's flat. In the clinic, of course, it goes like this. I think that's really worth emphasizing.
Yeah. So actually, one of the things we've been thinking about in the lab where I was a postdoc, Kishani lab at Harvard, is to ask, are there mechanisms in nature that actively select against resistance, right? So that's an alternative to trying to find new antibiotics. Can you find molecules that select against resistant and favor sensitive bacteria? And the lab identified several scenarios where this might happen. I mean, it's unclear whether they'll be relevant for the clinic, of course, but you can imagine such scenarios. And one very simple scenario is sometimes the degradation products of antibiotics can favor sensitivity and select against resistance, and that's because sometimes this degradation product is sufficiently similar in structure to the real antibiotics that they trigger the resistant mechanisms, in particular efflux pumps, and that's costly. So bacteria invest in resistance when it's not needed, and that selects for sensitivity. And it was to keep in mind that in natural environments, antibiotics spend most of they're quickly degraded and spend most of their time in the degraded state. So it's interesting to think that making an antibiotic, you actually create a net selection against resistance in the long term if you integrate over this entire time. So that's one mechanism. Another mechanism is antagonistic combinations of antibiotics. So it's usually in the clinic, people are looking for synergistic combinations, right? Combinations that amplify each other's effects. But imagine you apply antagonistic combinations that cancel each other's effect. And then think about the emergence of resistance now. If a bacteria evolves resistance to one of the antibiotics, it will not only stop seeing the antibiotic, but it will also eliminate the protective effect of this antibiotic. So actually, single drug resistant bacteria will be selected against in favor of bacteria that are sensitive to both antibiotics. So, I mean, of course, if someone is infected, you cannot apply these strategies, but you can think of in the clinic alternating antibiotic treatments with strategies that eliminate resistance and things like that.
In fact, you made the point that you think in the soil the antibiotics are actually not for controlling other bacteria, but for communicating, right?
Yeah, I mean, it's very fashionable these days to say they're for communicating. The key is to learn what they're talking about, right? And one maybe way to think about it is each of these antibiotic producers can make many antibiotics, and they have to decide which of those to make. So maybe they're kind of negotiating with some of these small molecules, so they're kind of doing diplomacy with it, and eventually certain antibiotics are made at very high concentrations. And actually it's also couples to these issues of cryptic secondary metabolism, which is also one of the hopes for saving us from this resistant crisis. So when people sequenced the streptomyces, which are the primary antibiotics makers in soil, they discovered that they can make 25, in some cases 40 different secondary metabolites, like antibiotic-like molecules, Whereas if you culture them in isolation in the lab, they make only 2 or 3. And then you wonder what are the conditions in which these cryptic metabolites are made. And if you can discover such inducers, obviously you unleash like 5 times more compounds for clinical testing than pharmaceutical companies tested before. And this is another way in which studying the ecology of microorganisms might play a role.
Right, right. It reminded me of, I think, quinolones, which are quorum sensing molecules, but we can make derivatives that are antibiotics, right? So they're communicators, but we have changed them to do something different.
We have, of course, because the fluoroquinolone antibiotics like ciprofloxacin, I mean, their history is that they actually emerged from the alkyl quinolone signal molecules that Pseudomonas makes, and that was the starting point for them.
Interesting.
Worth picking up on the comment you made about fitness and antibiotic-resistant bacteria. It's a fitness burden. That's a common perception, but bacteria acquire compensatory mutations as well. And if it was a fitness burden, we wouldn't have the global problem with antibiotic resistance. And, you know, bacteria really are older and wiser and cleverer than us. And, you know, if they want to survive, they will make sure they do. So what might be an initial fitness burden really doesn't last for very long. And in terms of multi-drug efflux pumps, really this is not a burden.
Okay.
So let's move on to you, David. And I have an email actually from a listener that's a good launch into this. We get lots of emails, and over time we read them as part of the show. This is from Clark, who writes, I understood, he had listened to an episode where we talked about the isolation of phages that could be used to treat acne. You probably know the paper. I understood that the major problem with using phages for disease treatment was due to FDA regulation. However, when speaking about acne, you suggested that there were ways for the FDA to approve them as a type of treatment. If this is the case, then why on earth aren't they being used to treat antibiotic-resistant TB and other such diseases? So maybe that's a good way to launch into what you do. I picked up your title, Regulatory Aspects of Phage Therapy. What is the problem?
That's why. Because basically, although there's nearly 100 years of using phages as medicines, they have to prove themselves in exactly the same way as every other drug. They have to go through the clinical trials process, phase 1, 2, phase 3, marketing approval. You can't just go out, pick up a batch of phages and put them on somebody's skin to cure their acne or their MRSA. Yes, there are phages out there against Propionibacterium acnes, no question. There are phages out there against Pseudomonas aeruginosa, Staph aureus, TB. But in order to develop them as drugs, you have to isolate them, characterize them, purify them, test them through preclinical models, then put them into the early stage clinical trials, then into the later clinical trials, typically into 2 large clinical trials, and then, and only then, can you seek approval. And with bacteriophages, no product has yet made it all the way through to market in the West. They are used in Eastern Europe and in a number of locations, but nothing has gone all the way through the process to market with the FDA in the United States or with the EMA in Europe. And it's only really when phages have gone all the way through that that pathway will be recognized as proven. And it takes time. I mean, Paul says we're 20 years away with quorum sensing. We're at least 5 years away probably with a phage drug, simply because of the necessity to go through all those stages. There might be ways to speed it up. Some people have been trying to do so, as I'll say in my talk, but those have been unsuccessful. They've gone to the European Medicines Agency and said, can you approve these things faster, please? There's a critical need, as was done with HIV drugs, and there's 100 years of data. But the European Medicines Agency have turned around and basically said, look, we don't make policy, we implement policy. Policy is given to us by the European Parliament, and they say they have to go through all the hurdles. So while I appreciate there have been a lot of antibiotic initiatives over the years, as you said earlier, they've been focused in on the infectious diseases community, the prescribing physicians. The people we really need to influence aren't the scientists, they aren't the medical people, it's the people who make the policy. And if you look at the representation in the UK Parliament, it's not a technocratic parliament. You've got a few doctors, a few scientists, but most of the people in there have backgrounds in commerce or law or journalism. And that's the people you need to reach out to. And I admire very much the work of Antibiotic Action in going and talking to lawmakers, because it's only by talking to lawmakers, getting pressure on them both from within and from the public, that we will get the changes that will push things through faster. And people are dying because of the lack of effective antibiotics. And you'd expect me to say this, but phages can cure diseases where antibiotics can't. I'm not speculating on that. We have done an initial clinical trial in chronic ear infections where the patients had been ill for up to 40 years in the test group. And patients were cured. 25% of patients cured within 6 weeks. One patient had been ill for 18 years, treated with a broad range of antibiotics, was treated with a 2.4 nanogram dose of phages, a billionth dose of conventional antibiotic, came back a week later, no symptoms, no bugs. I'm not saying it will happen in every case, but we know it can happen. But that is preliminary evidence from a phase 1/2 clinical trial. It now has to go phase 3, probably another phase 3, then seek marketing approval, and then get it onto the prescribing registers with organisations like NICE in the UK and similar organisations throughout Europe, for which you have to show it's cost effective.
Right.
I mean, the regulatory issues that you talk about are exactly the same for any anti-infective, and they're major challenges. And I know there's been moves in the United States, such as with the GAIN Act, to try and relieve the pressures. There's also been a lot of discussions at FDA and at the European Medicines Agency between companies and the regulators to say, how can we work together to get the trials right? Because the issue with, particularly antibacterial drugs is that, how do you get the right patients, the right numbers of patients? Because you're really making sure that your treatment, your medication is safe to use in patients. That's,
absolutely.
That is why the regulation is there. And the trouble is we don't have good measures to make sure the patients entering those trials for any type of treatment, phages or antibacterial molecules, we don't have the measures to diagnose which patients got what infection.
Well, in our case, we're targeting initially patients with antibiotic resistant infections who have been treated for some time, so we know what infection they've got. And you can go in and treat those patients, partly because there's a greater clinical need, there's nothing that can work on those infections, but also because you know what they've got. And if you can prove it in those ones and then spread out into the wider community, that gives you a way in. However, you're absolutely right. The regulators want safety before efficacy. The key is to prove that these things are safe. Now, phages have a long history of anecdotal use, but you have to do it within the context of a clinical trial that is fully regulated, double-blind, placebo-controlled, and that is what we are trying to do. And it's not just phages themselves that can be antibiotics, as we've shown in our trial. They can kill the bacteria, they can allow the body to heal itself once the bacteria are knocked down below a critical level. It's a form of biological control. But we've also got some very interesting work showing that phages can help existing antibiotics to function, both by opening up the biofilm and allowing the antibiotics to get in, and we've had a patent allowance in the US on this one, that you can actually apparently treat with phage and reduce the level of antibiotic resistance. Now, we're talking about evolutionary cost. It does seem to be, if you get the bacteria to take their eye off the ball to focus on the phage, they do lose some of the antibiotic resistance. And this opens up really interesting possibilities for using phages to drive antibiotic susceptibility, to open up the biofilms, using them in combinations. And this is a place where we're very active at the moment.
So you mentioned ear infection, treating ear infection. Is that what you're working on at the moment, or any others?
That's what we were working on with our phase 1/2 trial. That was never really just an ear treatment. The reason we went with ears is it is not ethical to do long-term chronic infections in animals. It's just not something you do. Mm-hmm. But pet dogs have a very similar infection to human ear infections. So we went into pet dogs and we did the dogs some good, and the owners were grateful and the dogs got better. And that gave us the preclinical to go into our first human work in ears, where there is a chronic ear infection caused by one bug, Pseudomonas aeruginosa, and we could treat that as our first in man. But we're expanding out from that. We're now looking at a number of other infections, topical infections, which you put the phage directly onto, which can be wounds, leg ulcers, burns, any range of things, and lung infections where you can actually aerosolise the phage directly into the lung. And particularly in cystic fibrosis patients, you have very high levels of bacteria in the lung, and phages like high levels of bacteria. That's lunch to them. When they get in, if there are lots of bugs there, you get lots of phages, because phages amplify, hence the name of the company, when they hit their target. So if there's lots of target, there's lots of phage. If there isn't, there's no phage, and it gets eliminated.
So one of the issues that came up when we discussed this was whether it would be necessary to tailor a phage to the particular patient, and if so, that would be a regulatory problem. So
again, I'll address this tomorrow. There are two basic ways to go. Pret-à-porter and sur mesure, they've been referred to by a Belgian group. Basically, one size fits all, a mix which you know is likely to be effective in most cases, which is analogous to an existing antibiotic, or having a bank of pre-approved phages from which you select a mixture for each patient. The regulatory position on that one is a lot less clear. It is less compatible with the Western model, but there are ways to address it, particularly, for example, with extremely valuable patients, such as our Australian colleagues are doing racehorses. Because if you take 6 months to cure a racehorse, you lose a couple of million in fees because they're out for stud. And in that case, you may well put in the tailoring aspect.
Right. Did I interrupt you? Sorry.
Well, only I was going to say, it's been suggested that commercially using phages in animals might be a very good route forward, because there's less regulatory hurdles to get over. But also, if you use them in animal food production, you could look to be reducing the numbers of bacteria that are foodborne, and that would be one area where you could say, well, phages should only be used in animals. I'm being deliberately inflammatory. Should only be used in animals because we're going to reserve things like antibacterial drugs to only use in humans.
There are, Paul, check me on this, how many antibiotic structures basically? About 2,000?
Gosh, was that many? No, I mean, you mean clinically?
Potentially available, the broad range of structures. Okay, let's call it 2,000, maybe I'm being generous.
Yeah.
On the surface of the planet, there are 10,000 billion bacteriophages. We don't need to reserve bacteriophages for any individual indication. We can go and find phages which have a different method of action for every individual indication. You might say reserve those for veterinary, but those over there can be used for anything else. Environmental disinfection, hygiene, human therapeutics. There isn't that tight limit that we have with antibiotics. So phages can be used in a number of indications. When I mentioned the phrase, there's a less regulation in the veterinary sector, to a friend of mine from a vet school, not a million miles from here, he burst out laughing. There really is a lot of regulation in the vet sector.
Yeah, there is, but you don't have the same issue over, when I meant regulation, the clinical trials issue to get a licence.
You don't, and we, for example, from our veterinary use of the phages that then went into humans, we were pretty close to a vet product. But what you do have in that setting is you run up against barriers, and one that we were talking to a veterinary company about was whether bacteriophages as an antibiotic would be regulated in the US by the USDA or the FDA, because the FDA is much more complex. Biologicals are regulated by USDA, antibiotics by FDA, even for animal use. And the answer came back, they're an antibiotic, FDA. And that stops you going first into veterinary uses, at least in the US, because you have to go through all the process for a much smaller market. Antibiotics tend to trickle down. They get approved for humans, and then they go down to animals, or up to animals.
No, they're different entities. They have to be different entities. So although they may be similar structurally to the bacterium, they may work in the same way, but they have to be different chemical entities. And there are actually thousands of different antibacterial molecules, even though there may only fundamentally be a few hundred basic core structures. But one of the issues that I just wanted to just address is that for an active phage preparation, you have to make it a mixed phage preparation to minimise selection of resistance, whereas with antibacterials, it's a homogeneous, it's a single drug. So obviously, with most seriously ill patients, say a trauma patient at A&E, you would automatically, if they were very ill when they were admitted, would give them 2, sometimes 3 antibacterials because you're life-saving. But with phage preparations, you would always give multiple phages in a single preparation.
Not always. Listex is marketed by a company, I don't know if I can say trade names, called Microbiology in the Netherlands. That is a single phage. It's
and
highly
does resistance emerge?
There's, apparently it's used for food uses, food disinfection, so it's not an evolving situation. However, generally, yes, all clinical trials, bar one very small one in Switzerland, have used mixtures of phages, very oddly referred to as cocktails. And most people use them. They use them partly because of the broader coverage. Phages, individual phages tend to be narrow, but you use a mixture to broaden that out. Also because that mixture inhibits the development of resistance if you select your phages, so they use different infection processes. So cocktails do seem to be the most popular approach. But one thing we've learned from HIV is going in with a single drug is not always a good idea.
Was there a question on Twitter?
Yes, there are a few questions through Twitter. One from Nicholas Tucker, who is asking if there are any worries about enhancing horizontal gene transfer when using phage therapy. I'll just ask a second one as well, and you can answer that. One from Connor Bamford asking, so what about phage resistance in bacteria?
Okay, two good questions. The issue of horizontal gene transmission is reduced by using obligately lytic virulent phages that cannot do anything but kill their target. There are two basic lifestyles that phages have. One is lytic, they kill. The other is phages that have the choice to do that or to integrate their DNA into the host genome or to leave it hanging around as an extracellular genomic element. That's called lysogeny. And phages that can elect to do that are called temperate. Phages that have to do that aren't really phages anymore. So it's the ones that can go lysogenic that can pick up and move around bits of DNA. A lytic phage is going to kill. An infected cell is going to die. It's not going to transfer its DNA. So at present, there is very strong pressure that the only phages that should be used are virulent, obligately lytic phages, which avoid that issue. And to the second question, there are a number of factors you have to consider when you're looking at phage, but do you want to restate the question again so I can make it precise?
Yes, sorry. So what about phage resistance in bacteria?
Okay. Yes, there is resistance to everything. Even people have been using metals. Silver has had a bit of a checkered history with the FDA, not just due to oxidation. And, sorry, chemistry joke there. And people have been using copper, silver, various metals because there's no resistance. But copper is widely used in agriculture as an antibacterial, and resistance is running at about 40%. You can get resistance to something as simple as a metal. So yes, you can get resistance to phages. The joyous thing is, as we showed in our clinical trial, you get resistance by a process of antagonistic coevolution. But if you get resistance to penicillin, streptomycin, copper, that resistance then stays. They are then resistant, unless they lose the genes, to that molecule. With phages, the individual phages can then change by a process of antagonistic coevolution and adaptation. It doesn't have to be genetic change, it can be phenotypic change. And the phages can counter that resistance. In our trial, we found of the 12 test patients, 3 patients were cured, 3 patients, when you sampled their ear swab, you plated it out, you put your original 6 phages on it, they didn't grow. But in every one of those patients, there was still phage growing in the ear. It had adapted, it had countered that evolution of resistance in a way that nothing else can. And we think that's a very exciting prospect.
You know, we could go on for quite a while, but I think we're running out of time here. So, yes, the nods. So we'll wrap this up. This episode of TWIM can be found on iTunes and at microbeworld.org/TWIM. And if you like what we do, one of the best things you can do for us is to go subscribe on iTunes and leave a comment or a rating there. That helps to keep us visible on the podcast directory and helps more people find us talking about this wonderful world of microbes. And we love to get questions and comments. I had one more that I don't have time to read you, but it is from a farmer who listens to us while he milks his cows. Okay? How cool is that? This is what we're doing with this podcasting. Anyway, send your emails to twim@twiv.tv. Laura Piddock is at the University of Birmingham. Thanks so much for coming back to do this. Really appreciate it.
Enjoyed it, thank you.
Paul Williams, University of Nottingham, thank you.
Thank you very much, too. Yeah, it's fun.
Kalin Vetsigian, I'm sorry, is at the University of Wisconsin-Madison. Thank you so much.
Thank you, Vincent.
I appreciate it. And David Harper is at Amplify Biosciences. Thank
Indeed.
you very much.
Thank
Good
you.
luck. I'm Vincent Racaniello, and you can find me at virology.ws. I'd like to thank the American Society for Microbiology for their support of TWIM, in particular Chris Kandian and Ray Ortega, who are here from ASM. Thank you guys for doing the live stream and the recording. And many thanks to the Society for General Microbiology for bringing TWIM to Manchester for the 2013 spring meeting. The music that we use at the beginning and end of TWIM is composed and performed by Ronald Jenkins. You can find his work at ronaldjenkins.com. Thanks for listening, everyone. We'll see you next time on This Week in Microbiology.